everyone. Welcome to today's webinar titled High Sensitivity, Targeted NGS Variant Detection of Viral and Bacterial Pathogens, a novel solution for healthcare and environmental surveillance and research. This webinar is part of the 10th Annual Genetics Virtual Week, and I'm Susie Valdez of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots and sponsored by QIAGEN. For more information about QIAGEN, visit them at QIAGEN.com. Now let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located at the right of your screen. You can also participate by submitting as many questions as you want during the presentation. You can do that using the Ask a Question box and just click Submit. If you have any trouble seeing this presentation, use the Help Desk button located at the bottom of your screen or use the lobby. I now present today's speaker, Dylan Barbera, a Global Product Manager at NGS Genotyping. For a complete biography of our speakers, please visit the Biography tab at the left of your screen. Dylan, welcome. You may now begin your presentation.
I'm here to speak to you all today about is our newly launched hybrid capture-based microbial panels for both viral and AMR testing. Just before we get started, all the products that I'm talking about today are intended for molecular biology applications only, and not intended for the diagnosis, prevention, or treatment of any disease. So we just recently launched 4 separate new hybrid capture-based microbial panels. We have a respiratory panel that is for 89 separate viruses, including SARS-CoV-2, influenza, rhinovirus, enterovirus, and many more. We have an AMR panel, antimicrobial resistance panel, that targets 2,786 unique AMR genes for bacteria. And it consists of targets that are pulled from QIAGEN CLC QMI antibiotic resistance database, which is a conglomeration of multiple different AMR databases that exist. We also have a viral sexually transmitted infections panel for the sequencing and detection of common STI viruses, which includes hepatitis V, HIV-1, and 19 different types of high-risk HPV. We also have an adventitious agent panel, which is for detection of 132 separate viruses, including SARS-CoV-2, influenza, rotavirus, and many more. Now, the adventitious agent panel is originally designed around doing a sort of quality control test that can detect contaminants that are problematic for different things that are manufactured in bioreactors, such as vaccines or gene therapies. However, it does serve as a good panel for the broad detection and sequencing of the 132 viruses that are in this panel. The content of all of these panels will be displayed in this talk today, but they're also available on the QIAGEN.com product page, where we have the actual list of the viruses that are targeted. Each viral panel targets the whole genome of each virus that is present in the panel, while the AMR panel is specific to the AMR gene that the targets are for. Now, these panels fit into the standard spot where NGS fits in QIAGEN's sample to insights workflow, where, as I'm sure you're aware, QIAGEN offers quite a few different DNA and nucleic acid extraction kits, and after that comes your NGS step. But what I want to highlight here is that there's 2 different routes of bioinformatics, where we offer, included with the cost of the kit, analysis for free on our GeneGlobe analysis tool, which is a browser-based bioinformatic analysis. And for a more in-depth analysis, we have built-in pipelines in our QIAGEN CLC Genomics workbench. So for the general steps and workflow for this kit, if you are starting with RNA or total nucleic acid, we include with the purchase of the X-Hive kit a reverse transcription kit that is a total nucleic acid workflow. However, if you're starting with only DNA, you can start off right at the QIAseq FX DNA library prep step. So what you get when you purchase the X-Hive kit is the RT kits, the actual hybrid capture panel and probe kit, as well as that gene globe analysis downstream. Something that is worth highlighting within this workflow is that in this RT step, we have built into the chemistry the QIAseq Fast Select ribosomal RNA depletion technology, which I'll highlight it over the next few slides of what that actually means for product performance. Also, I want to highlight the hybridization time of 16 hours, which is an overnight hybridization step. However, this does work out well for your general workday planning, where from already extracted DNA or RNA, the workflow from reverse transcription to setting up your hybrid capture takes, about one full workday, and then you would set up your hybridization overnight and come in and finish the next morning the rest of the library or the hybrid capture step. So as I mentioned, in our reverse transcription kit here, we have built in our QIAseq Fast Select technology, which means that you lose significantly fewer reads to ribosomal RNA, which plagues a lot of our competitor products. You can see in this slide here that the QIAseq X-Hive kit results in, a mere fraction of the reads lost to ribosomal RNA as compared to supplier T and supplier I. And what that means is downstream, you get significantly more unique map reads to target, which I will highlight in just a few slides. Now, you may be, if you're familiar with QIAseq NGS products, you may know that pretty much everything that we currently offer is amplicon based, except for our exome kit, which is also hybrid capture based. And so, there's a handful of reasons that we are expanding to use hybrid capture technology for this product. And really, hybrid capture is a lot better if you're trying to enrich for many targets at once. So we have two different SARS kits, the QIAseq SARS-CoV-2 primer panel and the QIAseq Direct SARS-CoV-2 kits. And those are both amplicon based, whereas this panel is for 89, the respiratory panel is for 89 separate viruses. And so that 89 full separate genomes is such a large amount of targets that amplicon sequencing kind of breaks down. And so hybrid capture sequencing is really the best method there. Now, luckily also, hybrid capture is less susceptible to dropout due to mutations such as amplicon-based sequencing. If you're familiar with the SARS, with SARS sequencing, you'll know that every time a new variant comes around, the question comes up of, does, this SARS NGS kit still work for this new variant of concern? And, with hybrid capture-based sequencing, the likelihood of that is much less. You may be familiar with the Arctic network SARS primer panels, where they're currently on V4.1, which is kind of the fifth version, actually, of the Arctic panel that they've had to redesign several times due to new variants of concern. And with a hybrid capture panel like this, you're less likely to have that sort of issue. And, these larger probes, allow for much more comprehensive profiling. Now, as compared to amplicon sequencing, hybrid capture does have a longer turnaround time. As I mentioned, there is a overnight hybridization step, whereas with amplicon sequencing, you don't usually have, full 2-day workflows like that. And, as a result, there is more hands-on time as well. But the cost benefit there of hybrid capture is worth it in that you can get so many more targets at once as compared to amplicon sequencing. Now I'm going to move on to some specific detail about our respiratory panel. So displayed on your screen right now is the 89 separate viruses that this panel targets. Now, again, I want to highlight that this list is available on the QIAGEN.com product page for this. and it will give you more time to go through this because there is quite a few different viruses in here. However, I want to highlight again that it includes influenza A and B, and SARS-CoV-2, rhinovirus, and it also has RSV in there as well as a few different coronaviruses that are associated with the common cold. Now, many of you may already be aware of, the importance of variant tracking of different viruses, but, in the past 2 years, the importance of doing variant tracking to determine, to keep track of viruses has become much more clear with SARS-CoV-2, where, we're trying to track and make sure that there's no widespread virus that has, moving towards immune escape away from the different vaccines that have been designed, and give us a little more lead time to design new vaccines if they need to be designed. It also helps, really track how a virus progresses through a population, and has been proven to be a useful tool in epidemiological research. So a big concern recently has been the effect of a SARS-CoV-2 and influenza co-infection, where there's a lot of active research in this area at the moment, where these panels will allow you to do detection of both viruses at once and characterization of both viruses at once. Evidence suggests that a co-infection of SARS-CoV-2 and influenza leads to up as much as a 10% mortality rate in patients that are infected with both of these. And, the difference in immunology for one or the other is kind of an interesting thing as well, where it seems that if you only have the SARS-CoV-2 vaccine and you get infected with both, it's not quite as protective of having just the flu vaccine and getting infected with both, where the mortality rate remains high. Something I want to highlight here with this graphic on the right is that influenza A is broken into 8 separate genomic fragments, which will have meaning for you when we get to data in a few slides from here. So this is going back to what I mentioned before with the built-in fast select technology to our reverse transcription. So the chart on the left is what I showed earlier, and the chart on the right is showing what this actually results in. So this data was developed with our QIAGEN Hive respiratory panel and was done on a SARS positive sample. So you can see on the left, again, that we have significantly fewer reads lost to ribosomal RNA. And what that results in on the right is significantly more unique map reads to target, where you can see as compared to supplier T that we have a little under 2x as many reads. And against supplier I, we almost have 2.5 times as many unique map reads to target. And what that means for you is that you get significantly better coverage compared to those panels. And, that allows you to do a lot better variant tracking as compared to those panels. Now, for a lot of different targeted viral sequencing, the only option at the moment is shotgun-based metagenomics, where you're doing whole genome sequencing of these viruses and looking only at, bioinformatically looking at your virus of concern. Now, this can be terribly expensive for reads on your sequencer, and also doesn't necessarily let you get as many reads as you want from viruses with low copy number in your sample. We see an average of 2,400x fold enrichment as compared to shotgun sequencing across the board with our viruses, with our targeted viruses, which is what this table is highlighting. You can see in light blue the amount of mapped reads when done with shotgun sequencing, and in dark blue is the amount of reads that we're getting with the actual targeted hybrid capture. And then the dotted line signifies the fold enrichment experienced on those specific viruses. And this is the kind of coverage and hybrid capture that you can see across all of these simultaneously. So just to give an example of what the coverage looks like here, again, these viral probe panels are targeted at the whole genomes of each of these virus species. So you can see here we have SARS, RSV, one of the coronaviruses associated with the cold, influenza A, adenovirus type A, rhinovirus 1A. This is just a selection of viruses that we can display here in a way that you can actually see. And you can see pretty broad, even coverage across the board for each of these viruses. Now, again, I had mentioned earlier that influenza A is broken into 8 separate genomic fragments, which is why you see this tailing off at these division lines. A classic issue with short-read sequencing is that as you get to the end of your genomic fragments, that you do see a drop-off in coverage, which you can see, at the end of each of these different 6 that are not influenza A, where it gets to the very end, there is a drop-off in coverage, but influenza A will experience that at the end of each of its 8 genomic fragments. Now, we have also done a fair amount of wastewater testing with this panel as well. There's a lot of interest right now in using wastewater for pandemic surveillance, and a lot of people have been doing that recently with SARS-CoV-2 specific sequencing. However, a lot of different public health labs are realizing, it would be great to be able to do pandemic surveillance beyond just SARS-CoV-2 because, it could easily be the flu or any other respiratory virus that becomes the next pandemic. And so what we have here are 17 separate wastewater samples that we use our hybrid capture panel on. And you can see a pretty broad detection of all of these different viruses displayed on the right, where you're able to simultaneously detect in these samples, these wastewater samples, SARS-CoV-2 if it's present, or influenza A if it's present, in any range of different respiratory viruses that are included in the panel. Now, not included in my data set today, but we also did some wastewater testing with our AMR panel, which we got some good data from. And we will actually be doing a separate webinar on April 5th that goes specifically into the AMR panel. And, from sample, we'll have participants there from our sample extraction team, our NGS team, as well as our bioinformatics team to go over the AMR panel and its different use cases, including wastewater. So moving on to the actual AMR panel. Antibiotic resistance is a growing health concern, that more and more people are becoming concerned with. In the United States alone, there's more and more deaths every single year of, from infections from previously treatable bacterial infections. And the US CDC has, established a list of bacteria and antibiotic resistance that it, believes to be an urgent threat, serious threat, and beyond different levels of classification. And with that, the United States CDC has, in just 2021, put aside $172 million for AMR research, as well as has established a network of labs that includes every single U.S. health department in terms of AMR research and AMR surveillance. There are similar initiatives in Europe and throughout the world in order to be able to track these infections and diseases. Because, a lot of people will refer to AMR as the next pandemic or the silent pandemic, as there are already many different antimicrobial resistance bacteria that are beginning to spread across the world. So our panel targets, as I mentioned earlier, 2,786 unique and separate AMR genes, which is across many different types of drug classes that it cause antibiotic resistance for. And this is a pretty comprehensive panel of the different types of drug classes and antibiotic resistance genes that exist. So to show what that means in terms of like phenotypes, as I'm sure you're aware, there are many different mechanisms for antibiotic resistance, where it could be, the cell develops an enzyme to degrade the drug, or develops a channel in the bacterial cell wall that will push the drug out of the cell, or maybe it'll mutate its enzyme that's attacked by the drug, or protein that's attacked by the drug, so that the drug no longer works. And what we're trying to highlight here is that, our panel is not specific to any one mechanism. We're really targeting all of the different types of molecular mechanisms that are covered in, that are affected by AMR. So what is the benefit of a targeted AMR panel versus just standard shotgun sequencing? So as I mentioned with the viral panels, a lot of the only method for actually doing any sort of NGS on AMR at the moment is shotgun sequencing, which it can be terribly expensive on in terms of reads on your sequencer, which means you can't multiplex many samples. If your population, your microbial population only has like a low rate of AMR, you might not be able to detect it. So with a targeted AMR panel, you're able to get a much higher sensitivity, which means that you can detect a far lower amount of AMR genes that are present in a sample. And it also allows you to guarantee that you actually capture your region of interest. So currently with shotgun sequencing, you might not actually assign enough reads to your sample that you actually are able to capture your AMR gene if it's there. And using a panel like this, you guarantee that you will be able to see it. And also, this allows you to multiplex considerably more samples, which will lower your sequencing costs downstream. So just to give an example here for the math, on a MiSeq with V2 reagents, if you are doing standard shotgun sequencing and you're trying to do AMR research, you may only be able to run 1 or 2 samples to be able to actually guarantee you see your AMR gene. With this kit, you'll be able to multiplex 14 different samples on a MiSeq with V2 reagents, which shows quite a large amount in multiplexing as compared to standard shotgun sequencing, which really saves you a lot of money downstream for your sequencing reagents. And with that, since this is a targeted panel and you don't have to do full de novo alignment of your whole genome, it is a faster data analysis turnaround time as well. So what I want to highlight here is kind of the difference in what you're doing with shotgun sequencing versus targeted sequencing. So this top track is standard shotgun sequencing, where you are amplifying the whole genome of the microbial sample. And so this specifically is a methicillin-resistant Staph aureus sample. And you can see here, you are covering a region that you're not necessarily concerned about, where you're looking for these specific AMR genes here and here, but you're getting a lot of reads here. And what that results in is a significantly lower coverage, where you can see the max level of coverage in the top track is 184. However, when you are only sequencing your genes of interest, you can see down here that there's a max coverage of over 9,000x. So when you're only targeting your actual regions of interest and not wasting reads on regions that you don't actually care about, you can get significantly higher coverage of the genes that you do care about, and then do a much better and higher level characterization of those genes. So what we have here is, I believe, 57 different saliva samples that we collected and did AMR analysis on. And in the top, you can see the different phenotypes that we were able to detect for AMR, as well as in the bottom, the different actual genes that we were able to detect for antimicrobial resistance. So you can see broad coverage across many different genes at once, and across many different phenotypes at once that were present in just general saliva samples. Moving on to our advantageous agent panel. So again, the actual list of the content of these panels is available on the product page and can be provided to you. But just to highlight a few different things in this advantageous agent panel, we have adeno-associated viruses, We have hepatitis A, B, and C, Epstein-Barr virus, norovirus. Somewhere in here is varicella zoster. But what I want to highlight in general is this is a pretty broad targeted panel, which is why we've seen a lot of interest in this panel, because it is targeted at such a broad different amount of viruses, where really for a lot of these viruses, your only answer at the moment is shotgun sequencing. To just go over again the definition of what an adventitious agent actually is, it's a potential biological contaminant that can be problematic in the manufacture of different bioreactor-created and manufactured things, such as vaccines, gene therapies, and antibody therapies. So right now, there is a lack of good detection methods of all these different viruses that are present in these samples, and there has historically been times when these types of viruses have gotten into things like vaccines. So there was a point that the PCV1, which is Porcino-Circovirus 1, got into the rotavirus vaccine, and SV40 got into the poliovirus vaccine. And so this is something that a vaccine got distributed and injected to thousands, tens of thousands of different people, and it actually had in it present a animal virus that wasn't intended to be there. So these pharmaceutical companies had unfortunately unknowingly been injecting these viruses into patients. And if that could cause a problem at the time was unknown. And now for these specific cases, it was determined that it wasn't causing adverse health effects on the patients. But the point is, with those, the content of the viruses in these panels, that you could easily accidentally end up with these viruses in your bioreactor batch. And it's not something that you want to risk putting into patients, because we don't necessarily fully know the effects. So, there is interest in having a bulk level testing of batches before, they're filled into vials, to make sure that they don't contain these viruses and things like that. And that is, at least bulk level testing kind of things that this panel can be done with this panel for actual detection. Again, similar to the respiratory panel, we see an average 2,400x fold enrichment as compared to standard shotgun sequencing across the viruses that are present or targeted for in this panel. And moving on to our viral sexually transmitted infection panel. Now, this panel is a bit smaller, but it does target hepatitis V, HIV-1, and 19 different types of high-risk HPV, including HPV-16 and HPV-18. Now, many people are already fully familiar with the effect of HPV on cervical cancer, but almost all, 70% of all cervical cancers are caused by both HPV-16 and 18. And, in recent history, semi-recent history, it was determined that HPV can integrate into the human genome, causing disruption in gene expression. When paired with QIAGEN's CLC Genomics Workbench vector integration tool, we can actually use this panel. And actually, for any virus that does integrate into the human genome, you can use this tool as well. But with the vector integration tool, we can determine where in the human genome that a virus has integrated and what genes have been disrupted. So on this slide, we're demonstrating that we can detect, using our viral STI panel, that HPV-18 has integrated into a handful of different sites in HeLa cells. Now, this is mostly already characterized, but it is a good highlight of the technology with our bioinformatics tools that this is possible. So for any of the viruses in the panels that I've already described that do integrate into the human genome, you can use this vector integration tool to detect where in the human genome that they have integrated. And there has been some recent research around the implications of HIV and HPV co-infection which is also somewhat of an interesting area of research, where it's being shown that having HPV can make it more likely for you to be able to acquire HIV, and vice versa, where having HIV makes it more likely that you will also acquire HPV. And, obviously either of these infections can lead to very problematic downstream diseases as well, which is why this area of research is something of interest recently. I wanted to highlight, the kind of coverage that you're seeing with HIV-1 as well in the panel. And so you can see here a genomic coverage plot that shows the average coverage that you'll see for HIV-1 across the panel. And in terms of actual ordering information, so what you get when you order any 4 of these panels is 3 separate boxes. So you receive the reverse transcription box, which comes with all the materials necessary for reverse transcription from total nucleic acid or just from RNA. And that is where that fast select is built in that I mentioned earlier. Now, if you go looking in this box for a tube that actually says fast select, you will not find it because it is built into one of the actual reagents that is used in the reverse transcription step. However, it is in there and does provide both of that depletion of RNA. Specifically, what's present in this panel is fast select HMR, which is for mammalian RNA depletion. And it also includes the HMR globin, which depletes globin RNA as well. Beyond that, you get a box with all the reagents for hybrid capture, which includes reagents at both -20 and 4 degrees. And after that, let me know if you have any questions.
Thank you so much, Dylan, for your time today and for your important research. Before we go, I also want to thank our audience for joining us today. And I'd like to thank our sponsor, QIAGEN, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by our speaker using the contact information you provided at the time of registration. And this webcast can be viewed for up to 2 years until April 21st, 2024. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everyone, take care. Bye-bye.